So the 50s and the 60s are when we really got to understand that central dogma of genetics, that DNA was transcribed into RNA and then translated into protein. Once we understood that whole process, the next thing to come is, well, how can we exploit that information? How can we use that to our advantage? And that's where recombinant DNA technology came in the era of DNA cloning. And so in the early 1970s, researchers discovered these enzymes called restriction endonucleases. These are enzymes that are produced by bacteria naturally. They're used by the bacteria to digest viruses that have infected the cell. That's pretty much their only defense against viruses. They chew them up so that they can't express their genes and replicate. But what we have found is that we can purify these enzymes from the bacteria, and then we can use these enzymes to cut DNA. And what's really exciting about it is the fact that these enzymes recognize very specific sequences, and so we can cut the DNA at known sites to produce reproducible sets of fragments. So once we can cut the DNA into pieces, we can use another type of enzyme called DNA ligase to actually paste the pieces together, and we can cut and paste DNA from different organisms and form what we call recombinant molecules. And so this is just a picture that's kind of depicting that process. So you can see here they have taken DNA from a human cell. So in this example, it's DNA for growth hormone, the gene for growth hormone. And then they've taken this hormone and they've cut it, or it's this gene for the hormone, and they've cut it with restriction enzymes to form what we call sticky ends. And we'll talk more later about how that whole process works. And then they cut from bacteria, they've also isolated a plasmid, which is just a kind of extra chromosomal circular piece of DNA that's found in bacteria. They carry kind of extra special genes like resistance for antibiotics and things like that. So they've isolated, again, we'll talk more about plasmids later on. But they've cut the plasmid with the same enzyme and produced the same sticky ends, so that now these two pieces can be joined together with ligase, and then this new recombinant molecule can then be inserted into the bacteria, and the bacteria is a host for this molecule, and then the bacteria will actually express this human growth hormone gene and produce that protein. And what's really exciting about that is, prior to this technology, the only place you could get human growth hormone to treat diseases in which people lack that hormone was to extract it from the pituitary gland of cadavers. And so now we're able to actually manufacture that protein in living cells. This is not a protein that we could produce in a lab synthetically by joining amino acids together. We just don't know how to do that yet. The only way we know how to make a protein is to let a cell do it using the old-fashioned way of transcription and translation, utilizing ribosomes. So, but by inserting the gene for the protein that we want to make into a single-celled organism like bacteria, we're able to make that protein. And so, like I said, the bacteria are considered hosts, and the bacteria also, not only do they produce the protein, but they also replicate that plasmid or that recombinant molecule over and over again. And so this process is called cloning, because you're making copies of the recombinant molecule. You can also use that cloning process to isolate specific genes and study their gene expression, do DNA sequencing, study evolution, as well as actually produce a product.
A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as EcoR1. The enzyme has a precise shape that allows it to run along the groove of the double helix, scanning, in the case of EcoR1, for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point. allowing a new piece of DNA to be inserted. When it cuts, ECO-R1 leaves a sticky end. This helps the new gene to attach. The joins are then stitched together by another enzyme called DNA ligase. The genetically engineered bacteria is grown in a culture medium. Very quickly, large numbers of the bacteria can be produced, each with a copy of the inserted gene. The bacteria duly manufacture whatever protein the gene codes for, and so the desired product is made.
So once they figured out how to do this, now it's just the scientist's playground. We've been able to utilize this technology in plants and animals in the food supply. We've been able to incorporate genes for resistance to herbicides, insects. We've been able to enhance the nutritional value of plants and food sources, and all those things have been introduced into our crop plants. So if you're ever driving down the road in eastern North Carolina and you drive this summer, if you drive by a field of corn, you might see a sign that says Syngenta or Monsanto. That corn has probably been genetically modified by adding a gene from bacteria called the Bt toxin that kills insect larvae. And so now these plants are inherently resistant to the insect, and we don't have to spray them with nasty chemicals to prevent, to keep the insects at bay. The genes that get transferred into the plants are called transgenes, and plants that have transgenes are referred to as being transgenic. So we can also do this with livestock, and in fact, in 1996, Dolly was the first sheep that was cloned by nuclear transfer. Basically, they took a cell out of a grown sheep and extracted the nucleus from that cell, and then put that nucleus into an egg cell where the nucleus in the egg cell had been removed. And so the Dolly was an exact replica, an exact clone genetically of the adult sheep that the nucleus was originally removed from. This is not something that's done routinely. I think it took 256 tries, something like that, for Dolly to be born, so that's not exactly efficient. It was more of a, let's see if we can do this. I don't think it's something that they would do routinely. But and there is thoughts of actually using livestock to produce drugs. So instead of getting them manufactured in E. coli or yeast cells or something like that, they would actually have the animals produce it and secrete it in their milk. And there are some drugs being manufactured this way. So we know the genetic basis for tons of different disorders. A lot of these genes have been mapped and cloned, and so, for example, like one we've known for a really long time, we've already discussed, which is the mutation associated with sickle cell anemia. And there's a whole lot more out there now that we're more familiar with. And this is a busy chart. It's in your book. It's just showing you a bunch of different diseases and which chromosome they're mapped to. Now that we know that these particular mutations can cause these specific diseases, they can actually do prenatal testing to see if you're a carrier for these diseases, and if, what the probability is that your children would also have a risk of getting these diseases. And parents now have the option to actually use in vitro fertilization to test for these disorders, and then to only implant healthy embryos. And it's called pre-implantation genetic diseases, or genetic disorders. Diagnosis, that's what it is. Yeah, pre-implantation genetic diagnosis. It's quite controversial, but it is something that's available currently. And in fact, you'll have an assignment to do this week associated with this lecture about some videos that you're going to watch that I think you're going to find very interesting about the way they're doing PGD. So there are a lot of ethical concerns. This is a slippery slope. You know, how easy is it to move from testing to see if someone has cystic fibrosis to testing to see how tall they're going to be or whether or not they're going to be athletic? You know, so these are issues that that we're struggling with now, and it's going to be interesting to see in the next decade or so how these issues are handled. And again, that's what the assignment for this week is all about. So just some terms. Genomics is the study of DNA, basically the structure and function and evolution of genes and the genome. The big part of it involves sequencing whole genomes of organisms. A good example was in the 90s when we had the Human Genome Project. This is a big topic in our recombinant DNA technology class. Proteomics is the study of the proteins that are produced. So what proteins are present in the cell, what are their functions, how do they interact with one another, you know, things like cell signaling. And then bioinformatics is how do we deal with all this data. So there's over 3 billion bases in the human genome. And so the Human Genome Project was a massive project that produced just massive amounts of data, and all that data had to be handled in some way. And so software and hardware, computer software and hardware had to be developed to process and store all this data. And that is the job of bioinformatics. And that's a very, very lucrative field. If you want to make a lot of money and you like working with computers, these people pretty much name their price because they are so valuable to the study of genomics. So the way that we used to study genes, and the way we're going to talk about it in the early chapters of this book, is we would first study phenotypes of mutations. So say, for example, you've got fruit flies and one has a white eye and one has a black eye, and then you would identify the genes that cause that phenotype. So you would like cross a bunch of white eyes with a bunch of red eyes and you know, go back and forth whether it's female or male and all this kind of stuff. And then you can, through the process, you can kind of figure out which chromosome the gene is on. And you can even, through these crosses, can determine if you're looking at more than one trait, how close are these genes together are they on the same chromosome? Are they on different chromosomes? Are they close together on the same chromosome? You know, you can actually figure that out. And so that's kind of the old way to map. You start with the phenotype and then you work your way back to the genotype. Now, in modern approaches, because we can sequence the DNA, classical genetics is somewhat outdated. It's obsolete. Instead, now we just sequence the gene, so we know the sequence. What we don't know is what the role is. And so we can, once we know the sequence, we have ways to remove that sequence, remove that gene from model organisms, and then we study the results and see what happened to that mouse. So if we remove a particular gene and that mouse you know, can't hear, then it must be something related to hearing. And so that's what we call reverse genetics, and that's more what we do today versus the classical methods. But we're going to talk about both methods as we move forward in this through this book. Okay, so like we were talking about the different classical versus forward modern genetics, and we talked about more model organisms like fruit flies and mice. So genetic studies rely on these model organisms. So they're used to study biology, and the results of these studies can be applied to other organisms because really we share our gene makeup is shared amongst organisms. We share thousands of genes with mice. We even share about 200 genes with bacteria. So what we learn about in a model organism, we can easily apply to ourselves. In order for a particular animal or organism to be a good model, it has to be easy to grow. It has to have a really short life cycle because we want to look at several generations, and in order to do that, you can't wait 10 years for, you know, the next generation to come about. In each generation there has to be a lot of offspring, because we're looking at this statistically, and in order for the statistics to be accurate, you have to have a lot of progeny. The analysis of the genetics has to be straightforward, and you know, it has to be easy to study. And so mice, fruit flies, corn for plants, Arabidopsis is another common plant. In your book, there's a list of model organisms, and it's table 1.1 that you can see here. These are some model organisms that are used to study specific diseases. So you can see E. coli, bacteria, Saccharomyces cerevisiae is a yeast organism, the D. melanogaster, that's a fruit fly, C. elegans, that's a type of nematode, D. rira, that's a, I can't remember what that is, and then the M. musculus is a mouse. Oh, I know, D. rira, that's the fish, the zebrafish, yeah. So Mendel set the stage for the study of genetics. Of course, where we are now compared to where we were just, you know, 120 years ago is just phenomenal. So we've learned so much in such a short amount of time, and you can see here the timeline. And what I love to tell students about genetics is the fact that some of the people that we would consider the fathers of genetics, like, well, of course not Mendel, but like Watson and Crick. Jim Watson is still living. He's super old, but he's actually still alive, and so that's just, to me, amazing. And it's interesting to get into this at the kind of the ground floor. Numerous Nobel Prizes have been awarded. And so now that we have all this information and we're learning about all this stuff, there's a ton of ethical concerns that come up that are just so fun to talk about. And it's just going to be, this is just a really exciting time to be studying genetics. And so, like I said, we're going to talk about some of those ethical issues in one of the assignments that you have to do this week. I think you're going to find it really interesting. So this is the end of chapter 1. So I'm going to end this lecture here. Make sure, you know, feel free to go through these videos again and get a good understanding of all the terms that are present in this chapter. And then when you feel ready, go ahead and move on to chapter 2.